Hello everyone and welcome back to yet another video from my channel, Interactive Education for free and the best education possible to the world, from the student to the student for a better experience. And today we're going to just finish up the topic we've been discussing for a long time now, that is cell cycle and cell division. And today we will be looking at the last topic of this chapter, that is cytokinesis. Now, let's have a look at cytokinesis. We've discussed in the last video that we basically have, and many other videos before in this unit, we've discussed that the cell cycle has two main units: interphase and M phase. Now we know that interphase is a preparatory phase, and we've looked at the three main stages of the interphase. We have the G1 phase, the S phase, and the G2 phase. And we've looked at the M phase. We know that it has two main phases, that is the karyokinesis, which mainly works towards nuclear division. And we also looked at cytokinesis, which looked at cytoplasmic division. So we have looked at this. We have looked at interphase, we've looked at the preparatory phase and all its subphases, we've looked at M phase, and we've looked at karyokinesis in detail in the last video. But now we have to discuss the last topic which will culminate this unit, that is cytokinesis. That is the cytoplasmic division. Although we had a short discussion on this, I did not discuss it in detail, so that's what we're going to do now. So let's have a look at cytokinesis. Until now, when we've discussed this unit, we have not actually gone into the depth of whether we're discussing about an animal cell or a plant cell, although we know they're a bit different from one another. So just have a look. Cell cycle, interphase, and M phase. The whole cell cycle is the same for all cells, provided that they're eukaryotic. Now, we're not discussing about prokaryotic cells here, mind you. We are only discussing about the eukaryotic cells. So for any eukaryotic cell, that's the phase, that's the procedure we follow. But let's just come to the last phase. In cytokinesis, there is a certain difference between how we go for how plant cells and animal cells perform cytokinesis. So there's a short or a small difference, right? Let's have a look. Let's first look at the animal cell. Now, again, let's have a diagram of the animal cell. So if I draw a diagram of the animal cell, of a general animal cell, you know, that has a nucleus and whatnot. So it basically just has a plasmalemma, that is the cell membrane, and it also has the nucleus. It does not have any rigid coating, right? So it's very easy for this cell to divide its cytoplasm, because we know that the cytoplasm is the fluid jelly-like structure, so it's anyway very flexible. And even if we look at the cell membrane, that too is flexible and very, very jelly and fluidy, and it is not rigid. So it is very, very easy for this cell to divide directly into its daughter cells, because there is no rigid cell wall or any rigid coating outside the cell. And let's have a look at how this actually takes place. Now, we've looked before, and we've looked at this before, that when we have an animal cell, you know that we have a cell which now at the end of the telophase has two nuclei. Right? We had the karyokinesis and we have two nuclei now. Now, how does the cell actually divide now into its constituent daughter cells, or into its daughter cells? Basically, the first step which takes place is the formation of, a kind of gap or a line kind of thing between just across the cell. There is a lining kind of thingy which is developing here, right? So this gap, or you may say this lining, or this cutting, or this, just we draw it across the cell, and it just divides across the cell into two parts, with each part with a nuclei. This particular lining is called the cleavage furrow. So the cleavage furrow develops across the cell, and this cleavage furrow ultimately divides the cell into two daughter cells. Right, so it's basically this cleavage furrow. It's going to divide the cell. It's going to break the cell. So you can see here the cell is being broken. The cell breaks. And once the cell breaks, we have these two daughter cells formed, right? That's how cytokinesis or cytokinesis takes place in animal cells. The easiest procedure, right? It's not too tough, is it? Well, now let's come to our very old friend, and I really don't like botany, so whenever this comes up, I feel really annoyed. That's the plant cell. Now, a plant cell is slightly different from an animal cell, and we've looked at the differences in our earlier classes. And we've seen that one of the major differences between a plant cell and an animal cell is that, unlike the animal cell, the plant cell is enclosed by a rigid coating other than the plasma lemma, or other than the plasma membrane, that is the cell wall. So it has a rigid coating called cell wall, right? And it also has a nucleus, right? That's the thing. So unlike the animal cell, the plant cell has this rigid cell wall, and it's not easy for the cytoplasm to easily divide into two separate cells, because again, this rigid cell wall does not allow for, a formation of two independent cells. So how will the cell divide? Basically, the best process to be followed, or the thing which actually is followed, is that a new cell wall is generated in between the cell or across the cell. So if you look at the animal cell, you know that we have a cleavage furrow which is forming. But if you look at the plant cell, here we have another cell wall which is forming. So a cell wall is formed across the cell, right? So as to, and obviously there'll be two nuclei, so there'll be one on each side. But how do we form the cell wall? How will we create the cell wall? That's the question. So let's have a look. Now suppose we have a plant cell, and after the, whatever your karyokinesis process, it has two nuclei on either side, right? We looked at that. So now we have a nucleus on each side. So one nucleus here and one nucleus right here. So we have two nuclei, right? Now we can formulate a cell wall here, just here, and this will be two cells right now. Easy peasy. But how do we do that? Now, if you have a look carefully, you know that the centrioles are basically, pulling away the chromosomes. Now, if you have a look there, now we know that we've looked at this during the anaphase of the karyokinesis, that during anaphase, they're just pulling away the chromosomes, right? The centrioles. These are the centrioles which pull away the chromosomes. And I've discussed this in the previous video. If you have not looked at that, I suggest you should look this unit in a sequence so as to help you understand each video, because it'll be really difficult for you to understand this if you don't have a look at the previous videos. So we have a centriole, and the chromosomes, and the centrioles are pulling up their webs in the form of the spindle fibers, which we discussed earlier, spindle fibers. And these spindle fibers have the kinetochore. The kinetochore combines with the centromere of the chromosomes, and it pulls away the sister chromatids, and we've all discussed that. But during this process, in animal cells, it's a very simple process. This takes place. But in plant cells specifically, at this process, the, what do you call, the chromatids, or yeah, you can say the chromatids, they start, releasing a particular kind of vesicle. Now, vesicles, they are tiny membranous membranous button-shaped structures, spherical-shaped structures which contain stuff. So that's vesicle. So they start, releasing some vesicles, or some vesicles are formed in between the cell. And ultimately, when you get this back in this form, right, when all these go here and they collectively form nuclei here, the centrioles and the chromatids, new chromosomes formed and chromatic material, so they form no nuclei, but still these vesicles which are present across the cell, across its middle, now these vesicles fuse. So these vesicles right here, they fuse with each other. The vesicles will fuse. And once the vesicles fuse, they form a, kind of another vesicle, a longer vesicle-like structure, a longer, this kind of structure, juicy, membranous kind of structure. And this membranous kind of structure formed due to the fusion of these vesicles is called cell plate. Right? It is called cell plate. Now, it is from this cell plate that the actual cell wall will develop. So some chemicals are secreted, and it's a very complex process. Some chemicals will be secreted. Ultimately, the cell plate develops. And once the cell plate develops, right, because these vesicles, after their fusion, the vesicles actually inside them contain, as we discussed, they contain stuff. In this case, they contain cellulose. And we know that cellulose is the material by which cell wall in plants is made. Right? Now we know that the plant cell wall consists of cellulose. So these vesicles contain cellulose, and after they fuse, their cellulose is released. And after a point, many polysaccharides, that is complex sugars, and all these things which are the main constituents of the cell wall, they come and just form a layer here, and ultimately this layer will form what we call the cell wall, which will be rigid and will divide these two cells. So if you see, that's what's going to take place. So a cell wall will develop, and due to the vesicles and the cellulose which is secreted, a cell wall will develop in between these two cells. And if you can notice here clearly, this will be the first daughter cell, and this will be the second daughter cell. So here, unlike animal cells, where the cytoplasm actually divides and gives rise to two proper cells, here you have just one cell which has two nuclei produced, and we are just dividing or partitioning the cell into two daughter cells. Since obviously due to the presence of the rigid cell wall, that will be easy, right? So that is how cytoplasmic division or cytokinesis takes place in the plant cell, and two daughter cells will be produced as a result. So with this, we conclude this unit, cell cycle and cell division. We looked at cell cycle, we looked at the two phases of the cell cycle, interphase, M phase, we looked at the different phases of interphase, we've looked at the two phases of M phase, that is cytokinesis and karyokinesis. We looked at karyokinesis in a lot of detail, and now we have also looked at cytokinesis, and we are done with that as well. So with this, we conclude this topic and this unit as a whole. It is actually part of the 11th standard I believe. I'm not too sure. It's part of 11th standard in CBSE, but it's very, very interesting, and it's somewhat related to the chapter reproduction in organisms, which is in class 10 as well as in class 12. And this will be the next unit I will be starting. Reproduction in organisms, a very, very simple yet important topic in biology. So with this, we conclude this unit. Thank you very much for joining me in this unit, cell cycle and cell division. I hope you had a lot of fun, understood a lot of things, and learned new things. Thank you very much. Do like and subscribe. Please do like and subscribe. If there are any comments, suggestions, or doubts, please post them in the comment section below. They are always welcome. Thank you very much. Goodbye. Stay healthy, stay smart, and do keep studying. Bye-bye.